Hello, everyone. I'm Marianne Laouri. Thank you very much for giving me the opportunity to kick off this session. That will be a very interesting session and only one more between you and the end of the day. So I am the global brand head for our most advanced product in diabetic retinopathy at Boehringer Ingelheim. And I'm accountable for the development, the approval, and the commercialization of this product worldwide. And I have a wonderful team that is involved in all aspects of the Mary Tyler Moore Vision Initiative and Consortium. So I want to thank all of them for all of their hard work and for all the members of the MTM Vision Consortium. Boehringer Ingelheim is a family-owned biopharmaceutical company, and we're committed to developing treatments for hard-to-treat conditions where there's a lot of unmet need, particularly among patients living with those conditions. We are deeply committed to innovation. But we know that even the coolest science is not going to reach patients unless there is consensus in the community about the value of that innovation, and it fits in to clinical practice and people have access to it. So because of that, we also believe in collaboration and not doing things alone, but to actually joining forces with many like-minded scientists, clinicians, advocates, policymakers, payers, et cetera, and other manufacturers. So when the opportunity presented us to join this consortium, we saw a very close alignment of the values of trying to stop vision loss from diabetic retinal diseases. But we also saw a call to action to try to change how diabetic retinopathy is being viewed today and to generate evidence of the natural history of this condition in order to be able to understand what are some risk factors and what are some predictors of progression, and how can we address patients' needs and work together to answer questions and fill knowledge gaps so that it will not only help us develop more meaningful and valuable treatments, but also help the community in their understanding of this at a time when resources are very scarce and we all share the same ambition. So as part of this consortium, we have been working on a few initiatives that you have already heard about today. Those are Protocol AR, which is sponsored by the DRCR, to understand the natural history of diabetic retinopathy, to develop a new staging method for diabetic retinal diseases, and to create new endpoints that are going to be approvable in order to expedite the development of innovative treatments for diabetic retinal diseases. We also are collaborating on developing a PRO to measure patient preferences and patient experiences with diabetic retinal diseases in order to make sure that the products, future products, address this. So in addition to that, we've expanded our collaboration to a lot of projects that we're doing with the University of Michigan and with the Joslin Diabetes Center. And I would like to invite Aude Couturier and Fabio Baschiera to join us.
So first, thank you, Marianne, for kind of depicting our efforts in collaborating, because as we have already all seen, resolution of any problem doesn't come from one item, one person, one group only. Good, so I really would like to kind of drive you through also one interesting initiative, which happened by chance, at least. So it's called the Bridge Study Group, and talking about the consensus in the selection of a group of structural and functional endpoints with clinical relevance in ischemic eye disease. And I'm presenting on behalf of the two co-chairs, Professor Siva Prasad and Professor Lowenstein. So disclaimer is obviously I'm Fabio Baschiera. This time I'm not Kai, I'm Fabio, head of clinical development for eye health in Boehringer Ingelheim. And the name Bridge comes from artificial intelligence. Obviously we want to be innovative, and it means building research innovation and developing global endpoints. So what is Bridge? Bridge is a study group of participants with the two co-chairs and 30 clinicians, mostly coming from Europe, and I'll tell you why. And these are the 30 ones, and it is a group also of other partners and collaborators, as we said. So we have Retina International, which is bringing the patient voice and the viewpoint, and this is where we start, and this is where we would like to end. The Meritar and More, obviously Meritar and More Vision Initiative, which is, we have already seen, is at the groundbreaking at the edge of innovation in a lot of those fields, especially in diabetic retinal diseases. And we have the Macular Society, which is a non-profit organization supporting patients in UK with macular conditions. And the innovative also part here is that we have 8 major pharma companies, as you can see the the logos here, working all together because we would like to achieve the same point. So where do we start? We started from the observation that from a European perspective, as you can see in the left-hand side in the A graph and the B graph, A is age-related macular degeneration and B is diabetic macular edema. These are the number of trials that are in the regions, that are happening in the region in 2024. So we are seeing constantly that in the FDA regions, the double of the trials have been conducted. And as well, so for both indications. So this is why we went together and we published the proceedings of that first meeting, and we located this to be the endpoint, the problem, right? So basically, we would like to come up with the output, which is an atrophic and ischemic retinopathies, so to look at clinical endpoints. And we haven't found a better name to kind of write atrophic and ischemic papers. Sorry, that's not a happy name, but starting anyway from the same structure relationship, ETDRS, early and late stage ischemia, structural methodologies, and then ischemia endpoints, functional methodologies, as well as the variables. And the structure of the bridge is 2 sessions of 45 minutes, chaired by the 2 co-chairs, and exploring the published evidences on those endpoints and the proposal for coming out with proposal of early indicators and correlation for structure disease. Today we're talking about ischemic diseases, and we are in the middle of the work streams. As you can see here, we are in the middle, and we'll finish by this year. And so let me get you through what the discussion is happening. So we focus on the ischemia variables that are predictive of disease progression. We have been through this discussion today a few times. And why that? Because ischemia is a recognizable pathophysiological status common to different retinal statuses. And obviously, the ischemia in diabetic disease, you can see in bold, can be related to the structural scale, to the ETDRS scale. So in the first meeting, we looked at the ETDRS scale, so I'm not going to go through it because I think that we discussed at length. But we looked at, with Professor Siva Prasad, she presented the retinal non-perfusion, the value of this endpoint and the unanswered questions. We know that DRSS score is associated with increasing RNP, so therefore there is a structure-function correlation, but there are a few issues with this measurement. So mainly, is there a threshold in retinal non-perfusion? What is retinal non-perfusion? If it is due to leukostasis or if it is due to endothelial cell death, one may be reversible, one may be not reversible. So 2 kind of retinal non-perfusion, as well as topographical location based on fluorescent angiography. So more questions than answers, and we'll come back to that. And the consequence is clearly to look at microaneurysms. What is the value and the challenges in measuring and quantifying microaneurysms? We're talking about endpoints. And we looked at, obviously microaneurysms are the first clinical signs of diabetic retinopathies. They increase with DR severity. So this is what we are looking for, a structure-function correlation, and maybe associated with neurodegeneration as well. But there are also challenges. The challenge is that microaneurysms and hemorrhages, a lot of the time, are grouped and counted together. So even their natural history is different. And small lesions sometimes are missed, and there is challenges in counting microaneurysms if there is edema. So definitely it will be using microaneurysm to look at treatment effect only microaneurysm may be a challenge. So another proposal of that we went through was from Professor Vujozovic here in the room, and looking into the disorganization of retinal inner layers and outer layers, so it's called drills and rolls. And the point, obviously, why this could be a good endpoint. For example, this is that absence or presence is detectable by OCT. Drills are associated with worsening of clinical visual acuity, and they are correlated with, again, with disease severity. So this is important. And the point is that drill may indicate, therefore, a kind of damage of photoreceptors and ganglion cells. So this could be another good endpoint. Now, retinal ischemia in diabetic retinopathy, when we're looking at it from a structure and function, so kind of going from the structure to the correlation, Professor Siva Prasad kind of brought us through the definition of the three outcomes when we are looking at microaneurysms, when we are measuring with fundus fluorescein. So microaneurysm, ischemia, and leakage. There are three characteristics that can be used together or separated. And we looked at the Ruby trial, at the Crimson, DRCR protocol AA to the INSPIRED study, and to the Vivid Vista, Clarity, and the Clarity, right, which is kind of a bit of a different kind of trial with PDR, but still ischemic index is a global measure of what can be retinal ischemia. Now, is measuring ischemia sufficient? Well, maybe not the only one. So, therefore, to one of the session, Professor Cunyavaz, who dedicated his entire life to look into do research on diabetic retinopathy, he kind of drove us through capillary non-perfusion and ischemia. So two aspects of the same medal, right? And through the OCTA quantification, we looked at the capillary closure as preferential, and preferential vessel shunts. We look at IRMAs, so intraretinal microvascular abnormalities, and their correlation with ETDRS scale. We looked at the association between IRMAs and ischemia, as well as the microaneurysm turnover rate, and also looked at the recent findings that hypoperfusion, especially in early stages, hypoperfusion is getting into hyperperfusion. So therefore, this is also an interesting, it was a kind of challenging topic. So in the end, this is what we are discussing and what we are going to discuss. Bridge results will be presented by Professor Lowenstein and Professor Siva Prasad. But a summary at the moment is that ETDRS represent a structural base of anatomical correlation of biomarkers of disease progression, and anti-VEGF reveal a dissociation between the structural and the functional outcome of treatment. Retinal non-perfusion is even visible before DR changes are visible, but the measurement may be challenging. Capillary non-perfusion is also an important topic when we are talking about DR progression, and ETDRS staging may be anticipated or may be enhanced in terms of diagnosis by other ischemia biomarkers. And so maybe is a composite endpoint a viable solution? We don't know. This is the group of Pharma contributors, and this is other contributors. And you may notice just in the middle, high up, a very interesting guy. And I'll leave you with that. Thank you so much.
Thank you. Very exciting finding. I guess I'm trying to think, we used to be very interested in pericytes,
yeah.
and which is critical for formation of microaneurysms. And one particular thing about the eye is that the loss of pericytes seems to be much greater than any other tissue. There are microaneurysms in the brain, but the ratio of endo to pericyte is much different than the retina, right? So I guess some question, any of the discussion as it becoming better and better on imaging technique, are there any technique to really go down to the cellular level, could differentiate pericyte rather than endothelial density.
Yeah, thank you. This is very interesting. I think that with Professor Cunha-Vaz, we looked at some OCTA data through kind of vessel density and vessel length, and through the skeletonized method. So, but not specifically to pericytes. I think that pericytes were a lot of the time. We looked into the theory of the pericytes, not the theory, but kind of the role of the pericytes in immature vessel formation, especially at the time when there was pegclaranib as a possible for Vista, when it was a possible treatment solution. But we have seen that actually it didn't make a change. You all may remember that there were ranibizumab or bevacizumab injection together with pegclaranib, 2 injection, 30 minutes by a side, distant, and pegclaranib didn't affect the pericyte formation, or at least the staging of and the outcome, the edema in DME patients. So it was pretty disappointing, I have to say. But since then, I don't think that we went through a lot into pericytes, rather than from a pathophysiological perspective. I'm not aware of any as an endpoint, but I don't know, maybe if there is anybody in the room that... Yeah, Stella?
Actually, there are some works recently published on the adaptive optics OCTA, evaluating the pericytes in diabetic retinopathy. Amani Favzi published that. So there are some works on the clinical evaluations of the pericytes.
Yeah, thank you, Stella. Actually, adaptive optics with transscleral illumination, I think, right? Yeah, this is actually an interesting technique to look at, cellular level of of what is happening in the retina. But I don't know. Thank you. I think it's...
I want to ask you a question about what made you and your colleagues in the Bridge Working Group join forces with the Mary Tyler Moore Vision Consortium. What are some of the advantages that we all get from joining forces? Can you speak to that, please?
Yes, okay. So this is definitely more philosophical rather than clinical, and I think that is truly important because first, diabetic patients are on both sides of the pond, right? So it started with the with an EMA-driven, with a European-driven, let's say, kind of study group, because we were worried that EMA is a bit immobilistic because we don't have NIH. Let's face it. So it is all left to the companies. And so in the end, we needed some enlightenment, also because Mary Tyler Moore, from a perspective, is definitely far more advanced. So honestly, we want to learn from Mary Tyler Moore. And Bridge Study Group is maybe limited in time, but definitely diabetic patients need endpoints beyond Bridge, I think. That probably Mary Tyler Moore is the answer.
Thank you.
Can I just add, am I on? Yeah, thanks. I just was going to ask Malvina, because the question comes up now, Fabio, because the work you're doing is extraordinary with Bridge, and it's quite encouraging for me to see all the different pharma companies kind of coming together on this issue, and it just reinforces and validates the positioning that we've established, that this is a necessary thing for us to all do together. Malvina has spoken to us about the need for creating this international standards on endpoints. So will we have an opportunity to learn from the Bridge and kind of incorporate that into creating these international standards on endpoints?
Yeah, no, absolutely. I think that the CCOI joining Mary Tyler Moore as well, I think that clearly gives the perspective, and it is important that we do collaborate with regulatory authorities to be at the table. So with Bridge, we're engaging with EMA only, but definitely we need to go beyond that, and I think that this is what is needed. So no, thank you. Actually, thank you for participating to that, and looking forward to the next steps. Good.
Any other questions?
I didn't want to follow Bob, but okay. So Lou Lesmes, AST. Fabio, on your last slide, you had composite endpoints as kind of like your last bullet. I was wondering if you could just give kind of just a high-level concept of what you think a composite endpoint might look like.
It's a very smart guy. Clearly got the secret there. So the secret is that clearly we're looking at a group of endpoints. But those group of endpoints, is it a group of endpoints what we're looking for, or one endpoint made by more endpoints? So cardiovascular style. So this, we don't know because we are in the middle of it, but it may be that we are ending up both for the atrophic macular degeneration, for the atrophic group, sorry, for kind of this work, and the ischemic work. I think that probably we may end up with both. It will be an interesting discussion with health authorities. It really depends on the inclination. If we have to have, from a statistical perspective, a list of endpoints, hierarchical, a number of endpoints that we are looking at, and let's say 3 over 5, or a group of endpoint that, it's either met or not met. So, and I know, Malvina, that maybe you're nodding. I think this is a typical question maybe for you.
Thank you. Yes. I think as far as a global consensus, it is a much safer way to go ahead and establish consensus on individual endpoints. Composite endpoints can sometimes work for a particular medical product, but I don't think a gold standard composite endpoint is a way to go. That gets very complicated, and it's going to create a lot of confusion for regulatory authorities. What we need is first, first of all, I didn't get the chance to applaud the incredible work the Bridge has done. I think it's fabulous, and I think, as you said, I echo your sentiment that joining forces is really going to get all of us forward, and I am a huge fan of Dr. Cunha-Vaz. And it was the work that EVICR.net has done for decades. We actually are partnering with EVICR.net in a different initiative. I think Jeff Goldberg might still be in the room, but was establishing for clinical centers of excellence. We're working with EVICR.net to come up with a global consensus on SOPs for measurements for variables that are being utilized in clinical trials, obviously including DRD. So that, and then endpoints is really the way to go.
Thank you, Malvina. I think I appreciated your kind of authoritative perspective.
Good afternoon, and thank you very much to Robert, Thomas, and all the MTM organizer for having me today. So it's my pleasure to share with you some insight about the EVIRED study, which is conducted in France. So I have no financial disclosure. So EVIRED is a multi-center study conducted in France. It has started in 2020 and it's still ongoing. It will end in February 2026. It has been led by Professor Ramin Tadayoni, who miss a lot. And it's a research program supported by the French authorities. You can see that we have academic partners. So the main one is Public Hospital of Paris. And also private partner, Zeiss for the imaging devices, and Atis Evolucare for the imaging transfer to the reading center mainly. So the aim of the EVIRED study is to develop an AI algorithm to help the clinician for the prediction of the complication of DR, so to assess more precisely the risk of PDR or DME at 1 year in a patient. So it's aimed to integrate all the data from the imaging, the more recent imaging technique, but also the clinical and systemic data that we collected from the diabetology department as well. So now we have 14 ophthalmologic centers, 18 diabetologic centers in France. We have already included more than 3,000 patients. The inclusion period has ended in December 2024. We have type 1 and type 2 diabetic patients, and there are annual follow-up for 2 to 4 years. all the patients have been imaged on 2 different devices for ultraviolet field color fundus photograph, OCT, and OCTA for the 2 eyes. The aim of having 2 devices at the same visit was to be able to develop an algorithm that is not device specific. So we have collected more than 5,000 images for the moment, and all the images are graded in the EVIRED Reading Center by 2 grader and 1 super grader, so 3 lecture of each image has been performed. The inclusion criteria were type 1 or 2 diabetes, adults. Diabetes duration needs to be more than 10 years for type 1 diabetes. We have excluded all the ungradable image for both, if ungradable in both eyes, history of vitrectomy in both eyes, and patients that already have complication in both eyes. So we were able to include patients with PDR or DME, but not with PDR and DME in both eyes, because we have nothing to predict in that patients. Here I will just present few preliminary results of the 1-year incidence of PDR and DME within the cohort. So for this analysis, we have included all the patients that already have complete 1-year visits, and which for the PDR analysis, we have excluded patients with PDR in both eyes at baseline or PRP or recent intravitreal therapy, anti-VEGF, because it can misdiagnose PDR. And we have excluded for the central DME incidence analyze patients that have central DME at baseline or also recent intravitreal treatments. So more than 8,000 1,800 patients have been included in the PDR analysis and more than 2,000 in the DME analysis. So regarding the baseline characteristics, in the 2 analyses, we have a majority of type 2 diabetic patients, a little more than 70%. The ABH1C was around 8%, so not very good diabetes balance. We have mainly patients with moderate and PDR, but you can see that we have almost all the stages that are represented. And almost 14% of the patients already have non-central DME at baseline. For the 12 months PDR incidence, we found that the overall was 2.67%, and it was, of course, differs between by baseline DRSS, so higher in patients with baseline severe and PDR, reaching almost 8%, while it was less in patients with mild or moderate and PDR. And among the patients that with PDR at baseline in one eye, the PDR incidence in the fellow eye was higher, reaching 10.8%. In the multivariate analysis, progression to PDR were statistically associated with the age, but also with the presence of neuropathy or PDR in the fellow eyes. Regarding the central DME incidence, we have overall incidence of 10%, and it was bilateral in almost 1/3 of the cases. And central DME incidence was higher in the patient with non-central DME at baseline, as expected, and reached 29%. It was also higher in patients with severe and PDR at baseline, and was around 20% in that cases. The factor associated was the type of diabetes, type 2, and the presence of non-central DME at baseline. So the result of our cohort are in line with recent results showing that the incidence of PDR and DME has declined these last years due to a better glycemic control, probably a better screening, and also maybe also anti-VEGF therapy. So with even if we have more and more patients with PDR and DME, because of the overall prevalence of diabetes, we have a lower risk for an individual patient to have an evolution to complication at 1 year. So within the EVIRED study, we try to develop tools to help the clinician to have a better assessment of the individual risk. One of the recent work of the AI team and my PhD student Hugo Leboité was to develop an automated segmenting algorithm to segment low quality areas on the wide field OCTA in this study. So we use Plexelite device images, and also to segmented the non-perfusion area automatically to be able to do this in a very easy and fast way in clinical practice. So we have developed and tested this algorithm in using 170 images from the patient of the EVIRED cohort with a very good accuracy. So we are now working on analyzing the whole cohort of the 3,000 patients to have the non-perfusion index over time in all the patient. In the first result that we have seen at 1 year, that there is not so many difference in the main part of the cohort at 1 year. Only patients with severe NPDR or PDR have a significant evolution of non-perfusion index at 1 year. What's next? So as I mentioned, EVIRED project will end February 2026. So we will have a few months for analyzing the data and be able to provide more insight in the results. But we also want to go on with the follow-up of the patient, and we hope to come very fast with the EVIRED Lift project, which will be with aim will be to go on with the follow-up of the patients that have been included in the EVIRED project, at least in some centers, to better analyze neurodegeneration, but also to add new functional testing as it will be made in the GRCNet study, so with contrast sensitivity, field analyzer, and ARG. So to conclude, in our practice today, the risk for one individual patient has been reduced. So we should have new biomarkers to better assess the individual risk of a patient to progress to PDR or DME. Probably AI algorithms are a promising tool to help in the screening, but also in the prediction of the complication of the disease. I wanted to thank you for your attention. Thanks again the MTM organizer for inviting me. And if you want to know more about our study, please visit our website. Thank you.
So any questions from the audience for Aude? Go ahead.
Sorry to ask so many questions. Very exciting. One US study showed, I guess I'm wondering if there are differences of DME or a PDR depends on the onset of your, looks like the bulk of your patients are type 2, right? On the age of onset of type 2, because the US study showed that in the youth with type 2 diabetes, their prevalence and the incidence of DR severity certainly is much more than just, over 40 age of onset, over 40 years of age. So
Yeah.
Did you see any differences in age of onset?
Yeah, I think you raise an important point. So the duration of diabetes have been shown to be a risk factor for PDR and DME development. In our study, the age of the patients, which could also be quite correlate with diabetes duration, have been shown to be correlate in the multivariate analysis to PDR at 1 year. It doesn't appear to be significant in the analysis for DME. What also can explain that is that we have also a specific population because the patient has been included in tertiary care centers. So it's mainly hospital-based ophthalmology ophthalmology department with usually more severe patients or patients that have already have a long duration of the diabetes. So it could maybe explain, because the overall duration is already long for the population, but this is something we will also look in more close detail in the future, and also with a longer follow-up.
In your in your multivariate analysis, you did show, or you said that there wasn't a difference with gender or with ethnicity, but what percentage of your cohort were non-white Caucasian? Like, did you just not have enough significance? And if not, then are you going to try and add some more to the cohort so we can look at differences?
Yeah, in fact, yeah, we have different ethnicity, but of course, the majority of the cohort, if I remember well, more than 60% of the patients are Caucasian. So it could be also one of the explanation. However, we are now looking at a more specific subgroup analysis in a department with different ethnicities and more variable, more different ethnicities. And during this subgroup analysis in the departments that are more representative of other ethnicities, maybe we'll show different results. But it's, of course, an important issue to address.
So I'm going to ask a question of Robert, I guess. So can you tell us, what prompted you to invite Aude and to include EVIRED in the broader collaboration with the Mary Tyler Moore Vision Initiative and the work that we're doing?
Yeah, thanks, Marianne. I think it revolves around the fact that Ramin and Aude had already started the recruitment for EVIRED when we started to think about our endpoint studies. So our first offer was to ask them to include the functional variables in the original study, so we could have visibility on the entire cohort. We weren't able to do that for a lot of logistical reasons, but we had gone through a very substantial process here on identifying promising variables and the devices that could measure those variables of function in the planning of our studies. We wanted to spread that knowledge to others who were doing longitudinal studies, to ask that they also consider those same sorts of variables in their studies, so we could just, increase the N of these longitudinal studies, because we're going to need more data in order to really validate these new variables. So it just so happened that with support from Boehringer Ingelheim and the device manufacturers that are here that are going to join the panel, we're able to get out the devices that we're using in our studies to Paris so that they can start to use those devices on the cohort that's already been followed for 2 to 4 years.
So I'm going to invite Fabio and our other panelists back to the stage. I'm going to ask you 3 questions, and you can pick, who wants to answer the first one and the second one, and then the last one will have to answer the third question. So introduce yourselves before you answer the questions, please. So you are 3 innovators, 3 device manufacturers who have decided to join the Mary Tyler Moore Vision Consortium. So I want to ask you, what value, the first question is, what value does joining that kind of a consortium bring to you in terms of the adoption of your innovation or being included in drug development and other, ways to advance our knowledge and options for patients living with diabetic retinal diseases?
I guess we'll start here. I'm Dina Dubey, CEO of LKC Technologies. This is the 4th year LKC has been here, so thank you, Robert, for the invitation. And we were impressed by everything MTM has done, the working groups, looking at all the solutions. And we're also a mission-driven company. We want to avoid any patients losing their vision. If it is preventable, why not? And we have an amazing technology, and this has been amplified very much. With MTM, all the connections, we you have all made and just throughout this room. That has helped. Our devices are in more trials than ever before, and the adoption continues to grow. So it has been a great privilege, and working with Jenny, Tom, for being the rock stars in their field. They're so open-minded. Thank you, and included in many discussions. So we're grateful.
Do any of you want to add anything to that question?
I'll add, just to kind of build on what Dina was saying. I think it was
Can you
Robert's.
introduce yourself?
Oh, I'm sorry. I'm Lou Lesmes. Lou Lesmes from AST. And so I've just been so privileged to learn so much, not only about kind of diabetic retinal disease, but retinal disease in general, from Tom Gardner, from Jenny, Lloyd Paul definitely taught me a lot. And the idea that then when the initiative came around and kind of Robert gave this call to action, it was one of those things where it kind of feels like you can't say no, right? You just say, where do you need us? And we'll be there, and what time and how many devices. And we were just felt like we had to make it happen. Again, just because we've learned so much from this community, and just as someone who started as a scientist, even kind of an accidental kind of entrepreneur, it was really the idea that there's an interesting problem. We may have a technology that can help, and so right, let's jump in and contribute what we can.
Well, thank you. We appreciate it. Do you want to add anything?
Hi, my name is Ian McMillan, and I head up sales and marketing for Cohen Medical, and I'm the objective field analyzer product manager. We're kind of here by accident. Because we didn't know we were going to be here when we acquired the technology 6 years ago, and here we are. This is my third symposium, so I've attended 75%, so that's a pretty good stat. So it's a privilege to be here, and it's hard to imagine this kind of an opportunity, both to be involved and also help. So.
Can you speak a little bit about the barriers that you face in having your technology be adopted in general, in the community and clinical practice in the U.S. and worldwide?
Well, we just started worldwide. We just got CE mark earlier this summer, and we started in the U.S. really in January. The biggest barrier is, in reality, practitioners don't really like to change. It's a paradigm shift, what we're doing. And doctors are interested, but they can be overwhelmed by something new. And so my job is to make it as simple as possible. So for example, the first thing I do is show them a report and say it's just like a visual field report that you're used to looking at. And then you can have a conversation. But it's a paradigm shift, and it takes a lot of getting used to. So we have our work cut out for us, for sure.
Yes. Right.
Sure, I'll go ahead. It's this team here shouting functional endpoints are important has been huge for all of us. This is what we go every day and tell the market, go to the clinicians. And we have to answer the question, so what, you do that, how am I going to change my practice? How is this going to change what I do for my patient? How long does it take? We cannot disrupt our flow. And that's something we have to look at in the trial. And I know we're having discussions on how long we're testing. And after all of this is done, what is it that we give to an average clinic who is seeing a lot more of the patients, right? Not just in research, not just in academic centers. How do we translate all of this? So it has been very helpful when the leaders in the industry are talking about functional endpoints, and all of us having devices that could potentially very much help with it.
So I'll actually take the industry perspective here,
Sure.
Because I think that this is clinical trials. Why, by helping patients, do we need the device manufacturer to come to develop endpoints with us? Because clearly clinical trials are the most controlled environment where you can set something that may be kind of widespread use in clinical practice be kind of applied. after we know what it does. So I think this is why clinical trials, and I think thank you to everybody for joining the industry and kind of helping us in doing our clinical trials, because this is actually when we are testing everything.
No, that's a good point. And do you want to say anything, since they're all part of?
Yeah, I think as a clinician, I really understand what you meant. I mean, we are not really refractive to change when we are a researcher, but when we look at clinicians that don't do research, of course, all will be new. It need to add a lot of value and to be very quick to be implemented in the practice, but also to have very practical consequence for the patient, either treatment or either a follow-up. So if we have like composite endpoints or with very clear functional structural correlation, saying if you have this decrease in this score, you need to have a 3-month follow-up or a 1-year follow-up is enough, or at least, since we don't have a lot of preventive therapy now, at least something to help to monitor the patient better. It will be, I think, easily adopted, but we need to have very clear guidelines or very clear endpoints to also adopt the technology, I think.
Would you like to say something? No? Okay. So I guess my last question is kind of the money question, in that, how do you think joining a consortium like the Mary Tyler Moore Vision Consortium is going to help drive the adoption of innovation? I mean, I think what we think at Boehringer Ingelheim is that even the coolest science is not going to reach patients unless, you really are clear about the value, where it fits in the treatment paradigm or the diagnostic paradigm, and how it adds value, and being able to articulate it in a simple way that people can understand. So this is what it's going to do to help me in my practice, and this is the value to my patients. And then the patients can also see what the benefit is to them, right? It has to be very kind of clearly articulated, not in baseball stats type language, but really something that resonates with all of us who may not be scientists or be familiar with the intricacies of the devices and the treatments. So can you talk about how that plays out for each of you? And Fabio and Aude, you can definitely join in that as well. Go ahead.
Yeah, sure.
Maybe I'll start, because as someone who we don't really market to the clinic yet, so I think I'll talk less about value and more about opportunity. And that credit to Fabio, credit to the consortium, that I know about 10 years ago when we took one of our prototypes to one of the pharma companies, and we talked about trying to get it into a trial and potentially validating our endpoint. They said, no, we need this, but the problem is if we validate this, then everybody else can use it. We kind of paused and we thought, oh dear, right? Like we're in the wrong place. And so thankfully we have someone like Fabio and Bob and the consortium now working, where they really see that this is a win-win, right? That when we validate endpoints, we can all use them, and we can do things like kind of solve the problems of retinal disease rather than solve the technical problems of endpoints.
Yeah.
So this is actually a very good point, Lou, because I haven't presented 3 times myself, so I think I don't do that.
No, you don't have to do that.
With different names, but that's okay. And the point is definitely that, in clinical trials, what you can have is the absolute value and the contribution to the patient. Now, the patient outside, and I would maybe comment on that, the real patient outside is not a clinical trial patient, right? Mm-hmm. So the clinical trial patient is highly selected, this is what the device can deliver, and according to what are the instructions, and kind of tribute to the technology people here. I think that clearly every implementation is bringing a new aspect to every device based on the algorithm or on any point. So I think this is really important. But I don't know if you want to comment.
Yeah, no, I fully agree. And I think for the patient, it's also really important to just also realize the impact that the disease can have on the visual function. And a lot of them complain, and we are not able really to understand why with only using BCVA. So having other way to monitor will be for sure helpful for the patient, but also I like personally to show to my patient the images and the results and some also quantitative analysis and to show the stability over time to reassure the patient, and that's also something very important in the relation with the patient. So I think it's really add also for the patient care in the daily practice.
But do you think that actually, for example, having a look at A wave and B wave is something that we need to upscale our clinical generic ophthalmologist to the higher level? Because clearly what we are measuring in clinical trials is highly specific. So having a maybe a report, what actually Ian was telling, but there needs to be a bit of kind of exploiting all the kind of specificities of what we are measuring. So I don't know, do you have any ideas on how to better kind of expand the culture of measuring new specific endpoints, which may be not easy to explain?
Well, this is actually part of the challenge because we measure a couple of different things that no other perimeter can measure. So that's an opportunity, but it's also a bit scary too, because that's the new part, right? So that all remains to be seen. And we're in a similar situation really where we're very, very new. So there's a lot that's to be determined. But so far in clinic, and I've been in a lot of clinics and tested a lot of patients, and although we don't have a huge customer base yet, the two things that are really promising are patients love it compared to not so much in terms of pushing a button. So patients like it. It's fast, it's bilateral, and those are all things that resonate with the patient. So that experience is really, really positive. And then from there, it's educating on the doctors on what these new results mean and to start to look at these new dimensions, these new measures that they've not seen before. So it's fun.
Good, go ahead.
Yeah, I mean, I'll just add, you can, you can have a lot of data. I mean, we're sitting on 250 plus peer-reviewed publications, right? And at some point, it's still not enough. But working with institutions, right, working with consortiums like this, that makes a huge difference in terms of it doesn't matter, what all you had. And that's where these conversations, these additional studies that are going on, all of that is incredibly, incredibly helpful. And the patient stories, I mean, that's the best part, for me coming here. And I was having a conversation with Candace. We just heard from another physician who had a patient like that and didn't see anything in fundus, I mean, had DR, but didn't see anything in fundus or OCT, but they had a very high score on Retival. And they decided to bring that patient back earlier. In 4 months, that patient had progressed to PDR. So it's one of those rapidly progressing patients you would not have seen for 6 months, 9 months, and you were able to catch early. So if you can catch early, make a difference, very meaningful.
Thank you. I just want to close, because we're out of time, by saying that one of the big benefits of consortia like this is something that Ali actually captured very nicely and said, when we all come together as industry partners and we say we're going to share, we're not going to act like the people who first said to you, if we get this device in our trial, then, we lose our competitive edge, right? But we really believe in transparency and openness and collaboration to move science ahead. Then we shame others into having to join us, right? To be able to be more transparent and to really make a difference. So thank you, Robert, Jenny, Tom, and everyone here who has made this possible for us to collaborate with each other.